Evening all, and welcome to our live seminar for B1, 2 and 3 before your OCR Gateway Biology exam tomorrow. So what we're going to do is in the next 90 minutes, hopefully, is run through as much of B1 to 3 as we can. Now, I do have the chat window up on a screen to my right, so I will be looking at it occasionally, but not throughout, because otherwise we're not going to get through this. And I'll try to cover as many things as I can. Obviously, don't start swearing things in the chat because that's completely unnecessary. So, it looks like we've got a fair number of people in now. It is 6:30, so let's make a start. So the first thing then in B1 that we need to know about is the fact that we've got two different types of cells. So you've got prokaryotic cells and eukaryotic cells. So the prokaryotic cells are bacterial and eukaryotic cells are the plant and animal cells. Couple of key differences between them are prokaryotic cells have no nucleus. They're very simple and they're the smaller ones, between 1 and 10 micrometers. Then our eukaryotic cells are more complex cells. They're between 10 and 100 micrometers, and they do contain a nucleus that has the genetic material inside them. So hopefully we do know our basics. Our animal cell, first of all, with our four parts: the nucleus, the mitochondria, the cytoplasm, and the cell membrane, making sure we only refer to a membrane in an animal cell. We've then got our plant cell, which has got a few more parts there. So we still have the nucleus, the cell membrane, the mitochondria, and our nucleus. But we also have the chloroplast, which is the bit that traps the light energy, the cell wall, the vacuole over there, and we need to remember those key differences between our plant and animal cells. So what we actually find, then we need to know what those parts actually do as well. So the nucleus contains the genetic material and controls the cell's activities. The cytoplasm is the site of all the chemical reactions. Our cell membrane controls what enters and leaves the cell. The mitochondria is the site of aerobic respiration, so it contains all the enzymes we need for that. The cell wall in our plants is made of cellulose, which makes it rigid and supports the cell. And our chloroplast contains the chlorophyll to trap light energy for photosynthesis. And the vacuole contains the cell sap because as it fills with water, it pushes out against the wall and helps the plant stand upright. Third type of cell, the prokaryotic cells. So again, we've got those key parts that we need to know on there, remembering that the ones underlined in red are the ones that are in all, and the ones not underlined only in some. So what we actually find is some of those key features in terms of our prokaryotic or bacterial cells is that we've got genetic material that is not contained in a nucleus. It's free floating in the cytoplasm. We've still got the cell membrane that does the same thing. We've still got the cytoplasm, same thing. The cell wall is made of a different substance called peptidoglycan. And again, it's all to do with protection of the cell. We've also got the flagella, which are the ones that allow the cell to move through liquids. We've got the pili, which are little hair-like structures on the surface that allow it to attach to structures and transfer genetic material between the cells. Some of them have this slime capsule to protect it from drying out. And we also have plasmids, which are little circles of DNA that store additional genes like that for antibiotic resistance. When we think about microscopy, then what we've got is our light microscope first of all. So make sure you do know the key parts on your light microscope, just in case they ask you to identify. So don't forget, we've got the eyepiece lens at the top. We've got the objective lens, which are the three we can switch between different magnifications for. You put your slide on the stage, and then we focus it with the two dials on the side, coarse focus and fine focus. Now, the whole idea of using the light microscope is to view small structures in detail. And it does that by passing light through the specimen from underneath, and then you can view through the eyepiece lens. If you want to know the total magnification, all you do is the eyepiece lens times the objective lens. One of the PAGs that you could be asked about is how to actually use your microscope. So we place our slide onto the stage, move it to the lowest position, select the objective lens with the lowest magnification, and then we're going to focus it, and then you can twist to the next objective lens and so forth. So this allows us to then zoom in and get the closer objective lenses in by not smashing the slide, which is always useful. Another thing they could ask you to calculate is the actual size of the image. So to do this, we've got magnification is your image size divided by the actual size. So that when you actually are given a diagram in your exam paper, get a ruler, measure carefully what they're asking you to do, and then all you need to do is obviously divide that by the magnification to get your actual size. One of the things we do have to do with our microscope is carry out staining, because many cells are colourless, so in order to make them easier to observe, we apply a stain to them. Now, depending on which stain we use, it might highlight the whole cell or a specific structure within there. 3 different stains we need to know about. Methylene blue, which is the one that highlights the nucleus in animal cells. The iodine, which is the one that highlights the nucleus in plant cells. And then crystal violet, which stains bacterial cell walls. Basics on applying a stain: you have your slide, you add a drop of the stain, and then we place the cover slip, which is that tiny piece of glass. Remember the name cover slip, because my lot were terrible at that previously. You put it on, tap it gently to get rid of any air bubbles, not just smashing it with anything, otherwise cover slip in teeny tiny pieces, and then you can view it under the microscope nice and easily. One phrase that we associate with our microscopes is the resolution. So what we find with resolution is the smallest distance between 2 points that can be seen as separate entities. So what we find is any structure that's smaller than 0.2 micrometers can't actually be seen with a light microscope. So to sort of get a new idea of how these cells actually worked, we actually came up with something called the electron microscope. And as a result of that, we can now see more detail. We can reveal greater information on our subcellular structures, and we can also understand the structure of tiny things like viruses to help develop drugs. When we're talking about the electron microscope, there are 2 types. We've got the scanning electron microscope, which allows us to see 3D images, and the transmission electron microscope, which just gives us the more magnified images. If you're asked to compare a light microscope and electron microscope, then our light microscope is cheaper to buy and operate. It's also smaller and more portable, but it's also got a lower resolution, whereas our electron microscope, the resolution goes up to 0.1 nanometers. For our light microscope, specimens can either be living or dead, whereas in our electron microscope, has to be dead. And the actual preparation of our samples for the electron microscope is very complex in comparison to the light microscope. So that's our first bit of B1, looking at cells and microscopes. The next section we move on to is looking at DNA. So hopefully you remember that DNA is found inside the nucleus in our eukaryotic cells, and there it's organized into structures called chromosomes. And those chromosomes can be broken down into small little sections of DNA called genes, and each gene codes for a protein. Key thing to remember is that everyone has unique DNA with the exception of identical twins. And what we find is that the proteins that they code for then give us all of our characteristics that are determined by genes. Even though every single cell, with the exception of your red blood cells, has a complete set of your DNA, then not all of them make every protein, because that would just be chaotic. So what happens is certain genes are switched on in some cells and off in others in order to actually make only certain proteins there. In terms of the structure of DNA, it is a double helix. It's a polymer. So what we actually have here are 2 strands which are wrapped around and joined by these complementary base pairs down the middle. So they're made of these individual monomers called nucleotides, and your nucleotide is made up of the deoxyribose sugar, a phosphate group, and one of the organic bases, adenine, thymine, cytosine, or guanine, or A, T, C, and G. So when they've actually twisted together in that double helix, the bit that joins those 2 strands together, those complementary base pairs, is A with T, so pointy letters together, and C with G, or curly letters together. So remember that phrase, complementary base pairing and double helix. For those of you doing higher tier biology, you need to know the process of protein synthesis, or how we make a protein. So it's a two-stage process. Step 1 is transcription. So this is where we're going to make a copy of our gene. So the DNA unzips in that particular region where the gene is, and then we've got these free RNA bases that come in and form complementary pairs with the DNA template. And we make something called mRNA, or messenger RNA. So once we've actually made the mRNA, it detaches once we've read that gene, and off it goes out of the nucleus where it's going to travel to a ribosome. So transcription is the making of the mRNA strand. Stage 2, again only for higher biology, is translation. So this is where our mRNA has left the nucleus, it's joined to a ribosome, and then as it passes through, it's read in sequences of 3 bases, or a triplet. So each of those triplets codes for 1 amino acid. So it's just a little code that tells us which amino acids are going to join together. So as the amino acid comes in that's complementary to that triplet, then the ribosome slides along, next amino acid comes in, and we form little bonds between each amino acid. And the sequence of your amino acids determines how the protein folds. So that is the second step, which is translation. Back to everyone again. The next thing we need to know about, which was always an old favourite, was the enzymes. So this used to be a good 6 marker on the old spec, so chances are it could come up again. Enzymes are proteins, first and foremost. So that means they are made of long chains of amino acids. So as a result, they fold up into this very specific shape. Now, the area that the substrate, which is the thing that's going to be involved in this reaction, binds to, is the active site. And the active site has a specific shape as the result of the enzyme's formation. One phrase we use with enzymes is the phrase biological catalyst, which just means it speeds up reactions in living organisms. Now, because we've got an active site that's a very specific shape, then that means it will only join with one substrate. So they're specific. And we can explain that using the lock and key hypothesis, that if you've got a key to a lock, it only fits one. You can't just wander into anyone's house because the shape matches perfectly. Same thing here with the substrate and the active site of our enzyme. So when the substrate joins with the enzyme, then we form the enzyme substrate complex. And then depending on the purpose of the enzyme, it will either break it down or join them together, depending on if it's building a larger molecule or breaking it into smaller ones. At that point, the products are released and the enzyme is then free to catalyze another reaction. In terms of what's going to affect the rate of these enzyme-controlled reactions, we've got 4 key factors: temperature, pH, enzyme concentration, and substrate concentration. So in terms of the temperature, first of all, then we get that very distinctive graph that starts off quite low and increases. So at that point, as you increase the temperature, you're increasing the kinetic energy of the enzyme, which means there are going to be more frequent successful collisions between the enzyme and the substrate. Therefore, the rate will increase up until we get to the optimum. Optimum is the fastest rate of a reaction. Beyond that point, if we keep heating it up, then the rate really drops off quite rapidly. And that's because we are denaturing the enzyme. So we're changing the shape of the active site so the substrate no longer fits. Second factor was pH. And again, what we see there is you get a bell shape here. So you've got your optimum pH, which will vary depending on where your enzyme is designed to work. And then if you go either too acidic or too alkaline from that point, the enzyme denatures, the substrate no longer fits the active site. In terms of the enzyme concentration, as you increase the concentration of enzyme, then the rate of reaction will increase because obviously there's more active sites available to carry this out. And if we increase the substrate concentration, it increases for a while, but then it levels off. And the reason that we get that plateau is because of the simple fact that you've filled every active site. But your number of enzymes remains the same, there comes a point when every enzyme is already working, so it won't increase beyond that. When we come on to our next topic of respiration, the first one we're going to look at is aerobic respiration. And the key thing here to remember is that the whole purpose of respiration is to give us energy. And every individual needs a different amount of energy depending on a range of different factors. So your age, your lifestyle, your gender, all of those will affect how much energy you actually need. Your metabolic rate is the speed at which the body uses energy. And the higher your metabolic rate, the more food you need to eat in order to actually maintain your body mass. So when we're thinking about the components of our diet, you've got carbohydrates, which are polymers made from simple sugars, which are used primarily for energy. So they get broken down into their monomers of the simple sugars by carbohydrase enzymes. Then we've got the proteins, again, polymers made up of amino acids, and the sequence of amino acids tells us what protein we actually have. We use proteins for growth and repair, and we break them down using a protease enzyme. Lipids are the third type we need to know about, which we use for energy, buoyancy, and insulation. And these are made from 3 fatty acids and 1 glycerol molecule joined together. And we break them down using a lipase enzyme. So what we find is as we break down those lipids into fatty acids and glycerol, then the pH will decrease because we're releasing fatty acids. And as the name suggests, they're acids. When we talk about aerobic respiration, then this is going to occur all the time to provide energy for all the different chemical reactions. So it takes place inside the mitochondria, which contain the enzymes that we need for this. And what we find is different cells in your body will have different numbers of mitochondria depending on their function. So muscle cells and liver cells have lots because they need lots. When we're talking about respiration for GCSE, we simplify it down to say that it's glucose plus oxygen makes carbon dioxide plus water. You do need to know the balanced symbol equation as well, which is C6H12O6 plus 6O2 makes 6CO2 plus 6H2O. Nice and easy one to remember how to balance it. It's 6 is in front of everything except glucose. Respiration is an exothermic reaction, so it gives out energy to the surroundings, and it's going to produce 38 ATP molecules. So aerobic respiration happens when there's plenty of oxygen, gives out more ATP. Anaerobic respiration is the one that happens when there is a lack of oxygen, okay? So we use this to transfer energy quickly and provide energy when the oxygen isn't available. So for example, if we're having plants growing in waterlogged soils, then they use anaerobic respiration. Now, what that means is when you get a lactic acid buildup inside your muscles, then you will suffer with muscle fatigue and you get cramps. So what we have is when you've finished exercising, you still have that increased breathing rate because we need to take in oxygen to then break down the lactic acid. So the oxygen needed for that is oxygen debt, which we have to repay. A second example of anaerobic respiration is fermentation. So again, we start with glucose, but this time we're going to make ethanol and carbon dioxide. So last bit of B1 is looking at photosynthesis. So hopefully we remember that photosynthesis is pretty much the opposite of respiration. So we start off with carbon dioxide and water, and we make glucose and oxygen. So if you've learnt one of those equations, you've learnt them both. Carbon dioxide will enter from the air through the stomata by diffusion, and the water enters from the soil through the roots by osmosis. Once it's in there, then what we have are photosynthesis taking place inside the chloroplasts. Just going back to one of the messages in the chat window there, 32 ATP is not correct. Go careful with what you read in a revision guide. It is 38 ATP in aerobic and 2 in anaerobic, giving you a difference of 36 ATP. So photosynthesis takes place inside our chloroplasts, and that's going to happen mainly in the leaves with a little bit in the stems. The chloroplasts contain a green pigment called chlorophyll, which is going to trap that energy from the sun. Photosynthesis is a 2-stage process. The light-dependent stage, which is stage 1, and the light-independent stage, which is stage 2. So for light-dependent, it requires the light energy from the sun in order to actually split the water molecules into oxygen and hydrogen. Then the light independent stage, step 2, this is where the carbon dioxide combines with the hydrogen ions to make the glucose. When we're talking about photosynthesis, it is an endothermic reaction, so it takes in energy from the surroundings in the case of sunlight. Once we've actually carried this out and we've made our glucose, we could use it in respiration, we could use it to make the cell walls because it's cellulose, we can use it to store food as starch, or we could store it in the fruit as sucrose, so we can change it into a range of different things. In terms of our actual experiments we need to know for photosynthesis, the main thing we need to know is how we test a beaker of boiling water for 5 minutes, ensuring that they are not above an open flame because that would just be a little bit hazardous, let's say. And then we wash the leaf with water, spread it out on a white tile, and then add a few drops of iodine solution to it. What we're going to find is whenever we're carrying out an experiment to determine what factor affects photosynthesis, then we have to carry out a process called destarching first of all. So the way we do that is we just stick the plant in a dark cupboard for 24 hours, because that means that in the experiment, any starch made is made in the actual experiment because the plant's used up any of its reserves. If we want to prove chlorophyll is needed first of all, then we get a variegated plant, which is one that's got some green bits and some white bits. And what we do is we destarch it, shove it in a cupboard for 24 hours. Then when we take it out, put it in the light for 24 hours, and then we test the leaf for starch. What you should find is only the green bits actually have the starch present because they contain the chlorophyll. If we want to prove light is needed, destarch your plant, then you cover part of a leaf with a bit of foil or a bit of cardboard, leave it in the sunlight for a few hours, and then remove the card and test the leaf for starch again. Third experiment is proving carbon dioxide is needed. Destarch the plant again, cover it with a polythene bag and add a pot of soda lime, and the soda lime absorbs the carbon dioxide from the air in the bag. Then we leave it in light for several hours, and what we find is we can test the leaf for starch and find out if we need carbon dioxide. If we want to prove that oxygen is given off, this is where we use our pondweed. So you put the pondweed in a beaker, collect the gas made, and then just use our test for oxygen. So a glowing splint will relight in that gas. When we're looking at investigating photosynthesis, then what we actually find is that we've got 3 factors we need to remember that will affect the rate. They're known as the limiting factors: light intensity, carbon dioxide concentration, and temperature. So if they ask you to calculate the rate, all you need to do is 1 divided by whatever time it took. For light intensity, then what we find is as we increase the light intensity, we increase the rate of photosynthesis up until a point where the graph levels off. And that point is when light intensity is no longer the limiting factor. If we look at carbon dioxide concentration, then the higher the concentration of carbon dioxide, the faster the rate of photosynthesis, until again the graph levels off when carbon dioxide is no longer the limiting factor. Third one, temperature. Is controlled by enzymes. So as the temperature increases, the rate increases as particles gain kinetic energy and therefore have more frequent successful collisions. And then past the optimum, we see it drops off quickly because the enzymes denature and the active site has changed shape, so it will no longer fit with the substrate. We generally use pondweed or Elodia for this because what we find is it gives off little bubbles of oxygen. So we can do a very simplistic experiment, counting bubbles, or we can collect the gas in a measuring cylinder to give us an actual volume. One of the things they could ask you is to actually evaluate the method of counting bubbles. Key thing there is just the fact that bubbles are all different sizes, so the volume won't be standard for each bubble, and it's really easy to miss them. So using the upturned measuring cylinder, far better in terms of accuracy. If we wanted to investigate light intensity, we'd have our pondweed, and then we'd move a lamp further away from it, recording the amount of gas made in a certain time. If we wanted to investigate carbon dioxide concentration, then again, using our pondweed, we just add different masses of a chemical called potassium hydrogen carbonate to the water, because that provides it with different amounts of carbon dioxide. And if we want to investigate temperature, we just place the equipment in a water bath and then run it at different temperatures to give us the same ability to compare. For those of you doing higher tier, either combined or biology, you need to know the inverse square law. So this is when you double the distance from a light source, the light intensity decreases by a factor of 4. So it's written as 1 over d squared, or 1 over the distance from light source squared. Whenever we're investigating a limiting factor for photosynthesis, then there will come that time when the graph levels off. And just remember, whenever you see that plateau or leveling off, it's because it's a different factor that is limiting it. So that is B1 in a nutshell. So we're still on track at this point in time. Next up is B2. So B2, first thing we need to know is diffusion. So diffusion is the net movement of particles from an area of high concentration to an area of low concentration. So they are moving down a concentration gradient. This is a passive process, which means that it's actually not requiring energy, and it will continue until the concentration gradient is 0. We've got 3 factors that affect the rate of diffusion. Distance, so the smaller the distance, the faster the rate of diffusion. The concentration gradient, a steeper gradient leads to a faster rate. And surface area, a larger surface area leads to a faster rate of diffusion. So, second one is osmosis. The definition for this one. The greater the difference in water potential, the greater the rate of osmosis. So what we actually find is if we think about our plant cells, then we've got 3 scenarios we need to know. So the first one is that as water is moving in, so we've placed it in distilled water, for example, then water moves into the plant cell and it will swell out and become turgid. If we were to place it in a very concentrated sugar solution, then water moves out of our plant cell and it will actually undergo this process leading to plasmolysis. So it actually pulls the membrane away from the wall. If we do the same thing with an animal cell, slightly different here. So if we put it in distilled water, water moves into the cell until it bursts because there's no cell wall to stop that. And that's a process called lysis. Whereas if we put it in a concentrated sugar solution, then it will shrivel up and become all crenated. Third process is active transport, which is the movement of substances from an area of low concentration to an area of high concentration. These ones are moving against the concentration gradient. So because they're doing this, then that means they need ATP. So that's going to give them the energy to do this. It's an active process. We also need carrier proteins in the cell membrane. So what we find is those cells that carry out a lot of active transport have a lot of mitochondria present to provide the ATP they need. 3 key areas that we're going to see active transport taking place. In the small intestine, where we're taking up glucose, nerve cells, which use a sodium-potassium ion pump, and in the roots of our plants through the root hair cells to pick up the minerals from the soil. Next process we're going to look at is mitosis. So mitosis happens in mitoses. It happens in normal body cells. So we're going to use this process in order to repair any damaged tissue, to replace any cells that have become worn out, and just to allow organisms to grow when we just need more cells. What we will find as a result of mitosis is that we have 2 genetically identical daughter cells. So we're producing a clone of the parent cell here. What we find in order to do this, we need a big process called the cell cycle. Now, the cell cycle is the whole process of cell growth and division that will occur. And we've got 4 key stages that we need to remember. There's the DNA replication, movement of the chromosomes, cytokinesis, and then the growth of the daughter cells. If we think about the DNA replication, first of all, then what we've got is the DNA is a double helix, if you remember. So it unzips, first of all, and we've got a single-stranded template. So 3 DNA bases then come in and pair up with their complementary base pairs, so A's with T's, C's with G's, until we've replicated the entire strand of DNA. Once that's happened, we get the movement of the chromosomes. So they line up along the equator or the center of the cell, and then they're pulled apart to the opposite poles of the cell. Once that's occurred, a new nucleus forms around the sets of the chromosomes, and then we undergo the process of cytokinesis, which is where that cell pinches in and splits. So we then have our 2 daughter cells. Each of them has a full set of the DNA. They're genetically identical. After we've had cytokinesis, then what we find is that the cells will grow. When they're fully grown, cell cycle starts again. In terms of another process we need to know the name of is the phrase cell differentiation. So this is where we're changing an unspecialized cell to a specialized one that carries out a particular function. So what we find here is that we will do this to allow our cells to become specialized, because that means that they're better at doing something. So it's things like sperm cells, fat cells, red blood cells. They've all become specialized to carry out their particular function. So make sure that you do know those specializations of those key cells. One type of cell that is not specialized, so it's an undifferentiated cell, is the stem cell. So if they ask you to name an undifferentiated cell, stem cell is all you need to write here. And what we find is our stem cells can actually divide by mitosis to differentiate into any type of specialized cell if they're from an embryo. So we've got 2 types. We've got the embryonic stem cells, first of all, which are, as the name suggests, found in embryos. They divide by mitosis and they can become any type of cell at all. So they can differentiate into any cell type. The adult stem cells, these are found in various tissues around the body, like the bone marrow and the liver and so forth. These can only differentiate into some cell types. So we use them to repair damage, but we won't be able to use an adult stem cell to make every type of cell in the body. It's not just animals that do have stem cells. Plants also have their own version of stem cells, and these bits are called meristems. So any part of a plant that grows has meristem cells. So we find them in the root tips, the shoot tips, and the buds. 4 key things about our meristem cells: they're small, they've got very thin walls, small vacuoles, and they've got no chloroplasts. When we come on now to think about transport around different organisms, then one of the key things we need to know is surface area to volume ratio. So the surface area to volume ratio is the surface area per unit volume of an object. So when we've got much bigger organisms, they actually have a lower surface area to volume ratio. So that means we actually get to a point when an organism can't get any bigger with just using diffusion alone, because diffusion would be too slow to supply it with everything it needs to survive. So instead, multicellular organisms have developed a range of adaptations to increase their surface area to volume ratio at those exchange surfaces. So that would be things like the alveoli in your lungs, for example, or the villi and microvilli in the intestine. One of the key systems that we use in animals, and particularly humans, for transport is the circulatory system. So your circulatory system is made up of the heart, arteries, veins, and capillaries. In humans, we have something called a double circulatory system, and that's going to be called such because the blood's going to flow through the heart twice on one circuit around the body, once through the right side and once through the left side. There's a big advantage to having this double circulatory system in the fact that we can have different pressures on the 2 sides of the heart. So hopefully we know one side of the heart only goes as far as your lungs, the other side has to pump blood all the way around the body. So by having those 2 sides separate, it means that we can have different pressures which will send the blood to the lungs and around the body there. It's also known as a closed system because the blood remains within the vessels the whole time. You do need to know how to identify the difference between an artery, a vein, and a capillary. So arteries, first of all, always go away from the heart. So A for arteries, A for away. And these are easily identifiable because they've got a very thick muscular and elastic wall. And the reason for that is they've got to withstand these higher pressures of the blood leaving the heart there. The second one, your veins, these ones have a much thinner wall and a larger lumen or space for the blood to flow through. And the blood is under much lower pressure here. So in order to stop the blood just getting so far and then going back on itself, they've also got valves. So your valve is just a little bit of skin that's in there that allows the blood to go one way, but forms a seal to stop it going back on itself. Third type of blood vessel, the capillaries. This is the site of the exchanges. So this is where it's going to exchange with the cells and the blood. Key thing about them is they're only one cell thick so that the fusion can happen faster. Now, in terms of some of our parts of the blood, we need to know 4 parts there. So red blood cells, first of all, the job of that one is to carry oxygen. So they've got a small size to fit through the capillaries, large surface area to volume ratio for rapid diffusion. They've got a special shape called a biconcave disc, so kind of like refreshers or Parma violets as the sweets. They've got a little dip in the middle, and that's going to increase the surface area to volume ratio. They contain hemoglobin, which is a carrier protein that binds the oxygen, and they don't have a nucleus. And the whole idea behind that is to leave the maximum space for hemoglobin possible. The white blood cells are the ones that are going to fight infections, and these do that in one of 2 ways. First one is they'll produce antibodies, and the second is they will engulf and digest a pathogen. Third part of the blood, the platelets, these help blood to clot, and this prevents too much blood loss, so you won't die from a paper cut, which is always a bonus. And it also seals any wounds to prevent the chance of infection, or at least lower it. The last part, the plasma, this is a straw-coloured liquid that pretty much everything else just floats around in, and it will also carry things like the digested food, hormones, antibodies, etc. When we come onto the heart, they're not going to give you an entire diagram of the heart and say, label it all. That would take way too long in an exam. They're not going to ask you to draw the heart because, again, this is not an art exam, this is biology. But they could ask you about some of the key parts. They could give you the diagram and ask what's part X. Or one of the ones that they did in the past was give an artificial heart and ask you to label what a part was. So if you know the flow chart, then this is going to help you out because you can work out where it's flowing and therefore what part that is. So if we start as the blood enters the heart, it does so through the vena cava, into the right atrium, through the tricuspid valve, so tri has RI just like right, into the right ventricle, then through the semilunar valve and into the pulmonary artery, off to the lungs where it picks up oxygen, then back through the pulmonary vein, into the left atrium, through the bicuspid valve, into the left ventricle, through the semilunar valve, and out into the aorta and around the body. Our heart has 4 chambers, 2 atria and 2 ventricles. Atria at the top, ventricles at the bottom. Think about the shape of the heart. It's got a little V at the shape at the bottom and V for ventricles at the bottom. It has those valves in there which prevent the backflow of blood, so blood only goes one way. The left side of your heart has a thicker muscle wall because that's got to pump blood all the way around the body, whereas your right side, thinner muscle wall because it's only going as far as your lungs, which are right next to the heart. And it also means that we can pump the blood at different pressures so that we can use a much higher pressure when going all the way around the body and a lower pressure when going to the lungs where we would damage the delicate tissues. Plants also have transport systems. It's not just us as humans. So when we're thinking about the plant transport systems, first one is xylem. So water is transported in xylem. Just go with the alphabet, WXY, water in xylem. And this is not only going to transport the water, but also the mineral ions. And it's going to take them from the roots up through the stem to the flowers and the leaves where it's needed. So water enters by osmosis and the mineral ions enter by active transport. Key features about our xylem: it's made of dead cells. There are no end walls between the cells. It's got thickened cell wall as a result of lignin being deposited, and that provides them with support. Second type of specialized plant cells are the phloem. Now, phloem tissue is used to transport dissolved sugars which are made in photosynthesis. So hopefully you remember photosynthesis, 6CO2 plus 6H2O makes C6H12O6 plus 6O2. And once we've made those sugars in photosynthesis, primarily in the leaves, we have to transport it around the plant in a process called translocation. Now, we're going to need to take those sugars to the meristems, where we're going to use them to make new cells, and also into the roots, where it can be stored for later. Key features about our phloem: they're made of living cells. They do have end walls, but these have become perforated and they are called a sieve plate, and those sieve plates allow the sugars to move through them. When we're thinking about the arrangement of our xylem and phloem, they're found in what's called a vascular bundle. So the vascular bundles are located in different areas around the plant, depending on where we're looking. So if we think about the leaves, then it forms a network with the soft tissue between them to actually support that soft tissue. In the stem, they're actually around the edge, which provides it with the strength it needs to grow vertically. And in the root, is located in the center to allow the root to bend as the plant moves. The next process we need to know is transpiration. So transpiration is the loss of water vapor from the aerial parts of a plant, or from the leaves if you prefer. And when we're talking about transpiration, go careful not to mix that up with the transpiration stream. So the transpiration stream is where we've got the movement of the water from the roots through the xylem and to the leaves. So transpiration is just the loss of water vapor from the aerial parts. Transpiration stream is the whole movement of the water. When we lose water from the leaves, we do have to replace it from the roots. So the plant needs to take up that water from the roots to replace anything lost at the top. Hopefully we remember that it's going into the plant by osmosis through the root hair cells, travels through the xylem, and then throughout the plant. When we get to the leaves, then we do have these little structures called stomata, which are tiny little pores, and that's where it's actually going to allow the water vapor to leave the leaf. And those are controlled by these guard cells, which are around the outside. So that when we've got plenty of water, the guard cells are all swollen up, and then they're nice and open for the stomata, so the water will leave. But when there's not much water in the plant, the guard cells close, the stomata, so it stops any more water vapor being lost. When we're considering what's happening in terms of pressure, then what we find is as we lose water from the leaves, then the pressure in the xylem vessel is reduced in the leaf. So as a result, then the water moves from the area of high pressure in the roots to the area of low pressure in the leaves, so it moves up our plant. The way we can investigate this is by using a bit of equipment called a potometer. So that's just a little bit with a gauge and a little tiny capillary tube where we put an air bubble in there. And then as the water moves into the chute and evaporates, the air bubble moves along the scale, and you can just measure that nice and easily. We do have factors which affect the rate of transpiration. Temperature, first of all. As we increase the temperature, water will evaporate faster from the leaf, which means that the transpiration rate will increase. If we increase the light intensity, then the stomata open in light. So that means that we'll have more water evaporating, increases the rate of transpiration, but it levels off again because we hit a maximum when all of the stomata are open. Air movement is our third factor, which basically just looking at the fact that when the wind blows, then it carries the water away from the surface of the leaf, which means we've got a steeper concentration gradient and therefore water will evaporate. With the humidity, which is the amount of water in the air, we see the opposite. So as we increase the humidity, we decrease the rate of transpiration because we've got a much lower concentration gradient. Right, that's now B2 finished. Still on track, which is good news. And B3 is our third and final topic for the night. So B3, first thing we're going to look at is the nervous system. So where we get a change in the environment, that's known as the stimulus. The stimulus is detected by a group of cells called receptors, and then we get a response occurring from the effector, which is a gland or a muscle. So muscles contract to move something away from whatever's causing the issue, and glands release hormones to have a response. When we're talking about the central nervous system, we're talking about the brain and the spinal cord. In order to connect everything around the body, we have three types of neuron. The sensory neuron, which go from your receptor cells to the central nervous system. The relay neuron, which connect the sensory to the motor neurons. And then the motor neurons, which go from your central nervous system to the effector. When we're considering what actually happens for a response, then we need to think about what's happening in a nervous reaction first of all. So we start off with a stimulus detected by a receptor. The receptor passes an electrical impulse through the sensory neuron, goes to the spinal cord and up to the brain in this case. Then we send a message back down the spinal cord through a motor neuron to an effector where we then get a response. And that's all going to take approximately 0.7 seconds. One thing we do need to bear in mind is the whole purpose behind having a brain. Now, the brain actually receives huge amounts of information from all these different receptors across the body at the same time, and it's going to process all of that information to provide a coordinated response. So this might result in a series of impulses being sent to different parts of the body to bring about a certain response there. We do have two types of response in our body. The first one, the voluntary one, so your decision to sit and type something in the chat, that's one. And you have the involuntary ones as well, such as your pupil contracting when you look at bright light. Now, reflex actions are involuntary and they take about 0.2 seconds, so they're much quicker than the ones we're actually consciously thinking about. So you need to know the reflex arc, which is stimulus to receptor, sensory neuron, into the spinal cord where it goes through a relay neuron, down the motor neuron to an effector where we then get the response. So make sure you do know the reflex arc because again, that used to be a bit of a favorite. For those of you doing GCSE Biology, then you do need to know about the eye. If you're doing combined science, you don't need to listen for a moment. So with the eye, we need to know those key parts. So hopefully we've all looked at this diagram and we know these key parts on here, and we need to know what they do as well. So the cornea is all about refracting the light to focus it. Then it passes through the pupil and is further refracted as it goes through the lens. And then the retina is made up of these photoreceptor cells, which produce a nervous impulse which travels down the optic nerve to the brain. If we want to focus on objects at different distances, then we need to change the shape of the lens. So the way we do that is by using the ciliary muscle. So if the ciliary muscle contracts, then the lens becomes more convex or fatter, and that lets us focus on objects up close. If the ciliary muscle relaxes, then it makes the lens less convex or thinner to focus on far objects. We need to know three vision problems: short-sightedness, long-sightedness, and color blindness. So if you're short-sighted, that means that objects in the distance appear blurry, and this is caused by the lens becoming too strong or the eyeball too long, and we correct that with a concave lens. If you're long-sighted, that means that near objects are blurry, and this is caused when your lens is too weak or the eyeball's too short, and we correct that with a convex lens. If you're colorblind, then it just means that you have difficulty making out the different colors, and this is a genetically inherited condition that usually affects males. Obviously, we do have two types of receptor cells in our eyes. We've got the rod cells and the cone cells. Cone cells detect the different colors. The rod cells work in low light intensity. Again, still for just the GCSE Biology group, here about the brain. Whole purpose of the brain in our head is to process all of the information that we collect from the receptor cells about both internal and external environmental changes. So it's not only going to take information from the nervous system, but also the hormonal system. And as a result of all of this information it's gathered, it then produces what's called a coordinated response. So using the brain as this central control center actually makes it a much faster process than if we had different little control centers scattered throughout our entire body. We do need to know all the bits of our brain there. So there's 5 of them we need to know, and we need to find them on the diagram because they could give you this diagram with a little arrow pointing to point X, and you would be expected to name the 5 parts. So 5 main areas of our brain: the cerebrum, which controls your complex behavior, learning, memory, etc. Cerebellum controls posture and balance. Your medulla controls automatic actions like your heart rate. The hypothalamus regulates temperature and water balance, and your pituitary gland releases various hormones. If we want to investigate which bit of the brain actually does what, then we actually have a range of different things we could use. So we can use brain mapping, which is using stroke patients and working out which bit does what. We can stick electrodes inside the brains of either animals or humans and see what happens when we send a little impulse down there. Or we could use the less invasive things of actual scans. So obviously people may not be too keen on having electrodes jammed into their heads for some reason, but we can use CT scans, MRI scans, and fMRI scans. So your CT and MRI will just give you images of the different sections of the brain. fMRI actually lights up depending on blood flow to different regions. So we can use that to actually give someone a task to do while they're being scanned to identify where is getting an increase in blood flow. A few key difficulties in terms of studying the brain. One, we've got to get consent from the patients because we can't just do this without someone's consent. And people might not necessarily want to give consent. If they've got some abnormality, they may not want to be viewed as one of the freaks in a freak show. We've all seen The Greatest Showman, kind of like that. Who wants to go and be stared at by people? We also need a lot of case studies in order to actually draw a reliable conclusion. And each study is actually an individual. So it's quite hard to draw very reliable conclusions from these. We can also find that many regions of the brain may be involved in a specific function. And if we're talking about animal testing, there's the age-old argument of it's not ethical to test on animals. If we're thinking about the nervous system, as we've said, there's the central nervous system, brain and spinal cord, peripheral nervous system, all the other neurons throughout the body. Now, if we have damage to our nervous system, it's damage to either the CNS or the PNS. Now, main causes of this, it could be an injury, it could be a genetic condition, could be a disease, or you might have consumed something that's just toxic. If we've damaged the peripheral nervous system, then this is generally going to be damage to your sensory and motor neurons, which results in things like an inability to detect pain or numbness. There is a limited ability to regenerate in the PNS, so minor nerve damage can self-heal. The bigger problem comes with damage to your central nervous system. Here we're going to lose control of body systems, maybe end up paralyzed, and this one can't regenerate. So it's going to be permanent unless we can correct it with some form of surgery. If we're looking at the problem of brain damage, number one, it's difficult to diagnose because we're looking at all these different things in our brain that, as we've said, have very key problems with actually investigating. If we're trying to treat it, then we've got a few options available to us. If we've got a tumor, then maybe radiotherapy or chemotherapy, maybe surgery to remove damaged tissue. But if it's in the center of your brain, then you've got to get through all the healthy tissue in order to get to the damaged region. Not an easy task. And we can also use what's called deep brain stimulation by sticking electrodes in to stimulate the brain function. Again, a bit risky. Sticking electrodes through healthy tissue might lead to damage elsewhere. Back to everyone, so combined biology, higher foundation, a whole lot of you again. The endocrine system. So this is where we're having a look at our hormonal system. So a hormone is a chemical messenger. They're made in the endocrine glands and travel through the blood in the plasma, as we've already discussed. Now, hormones will cause a specific response in target organs. Key differences between the hormonal and nervous system. Hormones tend to be fairly slow but long-lasting, whereas our nervous system is fast but short-lived. Hormones are chemicals in the blood, whereas the nervous system are electrical impulses which are transmitted along the axon of a neuron. Hormones will affect a larger area, whereas a nervous impulse will target a very specific area. When we refer to the phrase homeostasis, this is the maintenance of a constant internal environment. Now, what we find is that hormones are going to control processes that need this constant adjustment and balancing of the levels. So you do need to be able to identify where different hormones are produced in the body as well. So make sure you've had a look at that diagram. When we're looking at where they're going to have their effects, it's on target cells within the target organ. And the reason that they will respond is that they've got receptors on the surface that are a specific shape that matches that hormone. So once the hormone binds, then the receptor will produce a response in the target cell. If you're doing the higher tier, then we do need to know about the process of negative feedback. So negative feedback is very important in homeostasis. And what we actually have here is a cycle. So a small change in one direction gets detected by receptors, which then brings about a change as a result of the effectors to bring them back to their normal levels. So it's a little system where we're constantly monitoring and then having little tweaks to keep it around a very narrow level. One of the key examples of this is in terms of the thyroxine, which is produced in the thyroid gland. Thyroxine regulates the metabolic rate of the body, so it's all to do with providing energy. Now, the thyroid gland will take iodine from food and convert it into thyroxine by combining it with the amino acid tyrosine. So we start off with the hypothalamus, part of our brain, detects the low level of thyroxine in the blood. At that point, TSH is released by the pituitary gland, which travels through the blood to the thyroid, which tells the thyroid to make thyroxine. The hypothalamus then detects the normal level of thyroxine in the blood, tells the pituitary gland to stop making TSH, and as a result, then the thyroid stops making thyroxine. And it just keeps going around in that little loop of constantly monitoring and either releasing TSH or stopping releasing TSH to keep it all within the narrow limits. Second example is adrenaline, produced in the adrenal glands here and helps prepare your body for that fight or flight response. So do remember that when we've got adrenaline, it makes you respire faster, it increases heart rate, breathing rate, and it diverts the blood to the muscles. Back to everyone again now. So biology combined, higher foundation, whole lot of you back once more as we go on to the menstrual cycle. So the menstrual cycle is that monthly cycle that gets the body ready for pregnancy, and it lasts about 28 days. Female reproductive system, so the ovaries, the uterus, cervix, vagina, and fallopian tube. And what we find is that each month the lining of the uterus has to thicken to be ready to receive a fertilized egg. And at the same time that that's happening in the ovaries, then one of the eggs will mature. About 14 days after that, then it's released from the ovary in a process called ovulation. As it travels down the fallopian tube, if sperm is present, it may be fertilized. And if it's fertilized, it may implant into the uterus lining, where it's then protected, gets all the nutrients and oxygen it needs to develop into a hopefully healthy and happy baby in the fullness of time. If the egg's not fertilized, then the uterus lining will be removed from the body along with that unfertilized egg, and that's the period or menstruation. In order to control this, we've got 4 hormones which are made in the pituitary gland or the ovaries. We've got follicle stimulating hormone or FSH, estrogen, luteinizing hormone or LH, and progesterone. So first one, FSH, this is made in the pituitary gland, travels through the blood to the ovaries, and it causes an egg to mature. For those of you doing the higher tier, you also need to know that it stimulates the ovaries to produce estrogen. Second hormone, estrogen, is made in the ovaries and it causes the uterus lining to thicken. And when the estrogen levels increase, this inhibits the production of FSH. So that should stop more than one egg from maturing at a time. For those of you doing the higher tier, you need to know it also stimulates the pituitary gland to release LH. Third hormone, LH, secreted by the pituitary gland, and when the LH levels peak in the middle of the cycle, that's when ovulation occurs. Fourth hormone, progesterone, this is the one that maintains the uterus lining, and those levels will remain high throughout pregnancy in order to keep the lining thick. For the higher tier, you need to know that it also inhibits LH production. One of the questions that they could very well ask you here is to look at a graph. I say a graph, it could be a combination of 4 different graphs that are all stacked on top of each other. So do make sure that you can interpret graphs carefully by looking to see what's happening in each one in that same sort of line. So all you need to do to get these right is start off on the far left, have a look and see what's high, talk about that, and then move a bit further on to a change, and then look down all of the graphs to see what's happening and talk about them in turn. In terms of obviously stopping pregnancy, because obviously not everyone wants a baby all the time, then we have contraception available to us. So contraception is any technique that we're using to prevent pregnancy. So we've got two groups of contraception available to us. First one, the hormonal contraceptives. So these ones use hormones to disrupt the normal reproductive cycle. And the non-hormonal ones are barrier methods that basically prevent the sperm from reaching the egg. Now, in terms of the non-hormonal, first of all, you've got condoms, IUDs, and diaphragms. So they're all barrier methods which are preventing sperm reaching an egg in one way or another. In terms of the hormonal ones, we've got the combined pill or estrogen and progesterone pill, the progesterone only pill, and the IUS. So all of these are using hormones to disrupt that normal cycle to prevent pregnancy. Obviously, with each of those methods, there are positives and negatives. So make sure that if they ask you to evaluate the contraceptive techniques, look at the information they've given you in a question and think about what's good and what's bad. So thinking about the people taking them, will they remember to take a tablet every day? Will they be able to put them on? What's the effectiveness? And remember, if it says it's 99% effective, that means that the 99 out of 100 cases, there is no baby. So that's good. Don't get it the wrong way around and say that it's not effective. For those of you doing the higher tier, we need to know about how we can also treat infertility. So a few main causes of infertility. You've got blocked sperm ducts, not enough sperm produced in the testes, a lack of mature eggs made in the ovaries, or a failure of the ovaries to release an egg. So we can use hormones to treat some of these conditions, and that's our fertility treatment. One of those is using FSH as a fertility drug because that will stimulate the eggs in the ovaries to mature. We can also then have our estrogen production triggered, and that increases the chance of pregnancy. If, however, that's not going to work on its own, then we may well have to go down the route of IVF or in vitro fertilization. So in IVF, we're still going to use these fertility drugs to make lots of eggs mature at the same time. Once that's happened, we collect the eggs, as many as has been actually matured, place them in a Petri dish, and then we will add sperm to them. So the sperm and the eggs will then join in the dish. We'll check which eggs have actually been fertilized and which ones are developing properly, and then select one or two of those embryos to implant back into the mother's uterus. And from that point, it should progress as normal. There are, of course, some issues to do with IVF. First one, people argue it's not natural, and therefore you've got the whole religious arguments, etc. It does, however, give parents who wouldn't be able to have a baby in other ways actually able to have a baby. It means that older parents can have children, positive, negative, you can argue either way. It does have the potential for multiple births. And again, you can argue the positives and the negatives there. Obviously, if you're paying for it because it's very expensive, then positive, because multiple birth, buy one, get one free. It's always nice to have a little bob off offer in babies. However, the downside, if you've got multiple births, then you may have the issue of not being able to afford more than one at a time. For those of you doing just biology now, you need to know about hormones in plants. So when we talk about a tropism, we're referring to growth in response to an external stimulus. And this allows plants to respond to their environment. So we've got two we need to know about: phototropism, which is the growing towards the light, and gravitropism, which is growing towards gravity. So what we actually have to achieve this is a hormone called auxin. Now, auxin is made at the tips of the roots and the shoots, and it's going to stimulate shoot cells to grow more, but it inhibits root cell growth. So depending on whether we're talking about roots or shoots, different scenario. First one we're going to look at then are phototropisms. So this is where it's going to grow towards the light if it's positively phototropic, and away from the light if it's negatively phototropic. So what we see here is if we've got our little plant shoot here, Then at the top, in the tip, it produces the auxin. And the auxin diffuses down the plant and it will accumulate on the shaded side. So if the sun's coming from this side, then the auxin accumulates down here. As a result, this is a shoot, so the ones with auxin will grow more, so they elongate, and that means it bends towards the light. If we were talking about it being a root, the opposite would happen. It would bend away. Second, the gravitropism. So this is all to do with growing in the same direction as gravity. So what we find here is, again, the auxin accumulates, but it accumulates on the side that gravity is acting. So if we've got our actual root like this, and obviously that's down, so that's where gravity is acting that way, then the auxin accumulates on the lower side, and in roots what we're going to find is it's going to grow down, whereas in shoots it grows up. Still, just for the GCSE biology here, we need to know that auxins can be used to stimulate growth by causing that cell elongation. So this means we can regulate things in plants like fruit development. For those of you doing higher biology now only, then you need to know about two others. So not only do we need to know that auxins stimulate growth by causing cell elongation and can help regulate fruit development, but we also need to know that ethene causes fruit to ripen, and the gibberellins promote the growth, particularly in the stems, and we can also use them to end dormancy of seeds and buds. We do need to know about some uses of these different hormones in plants as well. First one is using auxins as a weed killer. So they're selective herbicides. So they will kill broadleaf plants, but not narrowleaf plants. So you can scatter that on your grass quite happily. The grass won't die, the dandelions will die. We can also use auxins in rooting powders, and that means that you can dip a cutting into this rooting powder and then the roots will grow. We can also spray auxin on trees to delay the ripening of the fruit, which means we can collect the whole harvest at the same time and prevent fruit dropping off the trees and becoming damaged. Fourth one is we can use ethene to spray on fruit trees and plants to ripen the fruit quicker so that we can get it ready earlier in the growing season for sale. We can use auxins to actually apply to unpollinated flowers so that they produce seedless fruit, like seedless grapes, and that's parthenocarpy. So what we find here is that we can actually carry out that pollination process without actually pollinating, which is quite nice. Obviously, we can use the gibberellins as well as auxins here to trigger germination. So if we've got seeds that we want to grow out of their season, we can apply auxins or gibberellins to trigger germination so we can grow them all year round. For just GCSE biology now, but both foundation and higher tiers, hopefully we do know that normal human body temperature is about 37 degrees Celsius. And the reason for that is that the actual enzymes that are inside our body all work at that optimum temperature of about 37. So we do have problems if our body gets too hot or too cold. So we need to make sure that that doesn't happen. So that what happens there is you've got these receptor cells in your skin that monitor the external temperature. And we've got internal receptor cells that monitor the temperature of your blood. All of that information is sent to the thermoregulatory center in your brain. And as a result of that, it will send impulses to effectors to make sure that the temperature is kept within that narrow range. So this is a good example of homeostasis. So if it's too hot, then the hairs on the body lie flat. And that's good because it prevents any air being trapped, and air is a good insulator, so we don't want that if we're too hot. Sweat glands will produce sweat, and then as the water evaporates from the surface of the skin, it transfers energy from your body to the surroundings, cooling you down. And we have a process called vasodilation occurring as well, which is where the blood vessels supplying the capillaries near the skin will widen, and that increases the blood flow through the capillaries, so you lose more heat via radiation. If you're too cold, then what we find is the body hairs rise up, traps a layer of air against the skin, which insulates your body quite well. Your sweat glands stop producing sweat because you don't need that. And we have vasoconstriction, which is where the blood vessels actually narrow to reduce the blood flow and therefore heat loss. One other thing you can also have is shivering. So that's where your muscles are contracting and relaxing really quickly, which means respiration happens and we generate extra energy. Back for everyone, biology, combined, higher, foundation, whole lot of you again, controlling blood sugar. So what we actually find is during digestion, then we release glucose from our food. So that's got to move from the small intestine into the bloodstream, and then we need to do something with it. It's not good to have vast quantities of sugar circulating through your body the whole time. So we can use it as an energy store, and the way that we're going to do that is by transferring that glucose into a different chemical. Now, what we do find is that if we don't transfer that glucose and we have this large quantity of it circulating through our blood, then we can have damage to things like your blood vessels and nerves, etc. So the way that we control this is inside your body, you've got the pancreas, and when the blood sugar levels are too high, it releases insulin, which is a hormone. That insulin travels through the blood to the liver, which is your target organ, and it triggers the conversion of glucose into glycogen for storage. So insulin causes glucose to change to glycogen for storage, bring your blood glucose level down. For those of you doing the higher tier, you also need to know what happens as an inverse to this, so that if your blood sugar level drops too low, then what happens is you release a hormone called glucagon, and that converts glycogen into glucose. Go careful here. There's a lot of very similar glue words. Don't mix up glycogen, glucagon, and glucose. If you've got an inability to control your blood sugar level, then you suffer with diabetes. So 2 types. Type 1, which is where you can't produce insulin because your immune system has destroyed the pancreatic cells that make them. And we normally control this with insulin injections. Type 2 is where we can't effectively use the insulin, or we're not making enough. So this one we can mostly control with a balanced diet and exercise, but sometimes it will also have the need for insulin injections. For those of you doing GCSE Biology, then you need to know how we control our water balance. Hopefully we know water will enter our body as the result of food and drink, and also be made in our cells as a product of respiration, and we'll lose it from the body in terms of sweat, urine, and exhalation. So when we're talking about the process of excretion, that's where waste products are removed from the body. So if we've got too much water present in your blood plasma, that's bad news because your blood cells could burst. And if we've got too little water, then what we find is the blood cells will start to shrink as the water moves out. Neither is a good situation, so we need to control this. If we've got an excess of water, a lot of it ends up in urine. So your urine is not only water, but also urea and other waste substances that need to be removed from the body. And the key area that's going to remove chemicals like urea, which is toxic, are the kidneys. So what we find is your kidneys will remove the urea. It then trickles down into the bladder where it's stored until you're ready to go to the toilet. If you're short on water, then what we find is very little urine is produced, but it's very concentrated. Whereas if you've got a large surplus of water in your body, then your kidneys will produce a lot of very dilute urine. In terms of the kidney, we do need to know the key parts of it. So we do have 3 key sections: the capsule, which is the outer membrane there, the cortex is the outer part of the kidney, and the medulla, the inner part of the kidney. So make sure you know those different bits on a diagram. When we're thinking about the actual structure within the kidney, we have these things called nephrons. Now, the top of the nephron is in the cortex, and the lower section is in the medulla. So blood enters the kidney under high pressure through the renal artery, and then it leads to this thing called a glomerulus. Now, that's basically a knot of capillaries. So what happens there is, as it enters the glomerulus, then as it tries to leave, it's a very narrow exit. So blood pressure is increased in that ball of capillaries. So as a result of that, it forces those small molecules like water, glucose, urea, etc. out through the capillary wall and into this structure called the Bowman's capsule, which has the glomerulus inside it. Anything larger than that, things like the blood cells, etc., they're too big to fit through the capillary wall, so they stay in the blood. So that first stage is ultrafiltration. Then, as the filtrate moves through the nephron tubule, all of the glucose is reabsorbed, as well as some water and any salts that our body needs. So because it's only reabsorbing certain things the body needs, that's the selective reabsorption. From that point, the filtrate then passes through the loop of Henle and the collecting ducts, which is where extra salt and water are reabsorbed if needed. If you're doing higher tier biology, you need to know what controls this, which is a negative feedback system. So this uses another hormone called ADH, or antidiuretic hormone, and the ADH actually makes the walls of the collecting ducts more permeable to water. So again, make sure you can talk about that flow chart of how ADH affects your water balance. Still, for the higher biology only, what we find is that if you've got a reduction in water potential of your blood plasma, then your body's thirst response is triggered. So that basically means an impulse is sent to your brain telling you, have a drink. That also means that the kidney is going to be making less urine. If you decide to ignore that signal going up to your brain telling you, have a drink, then what's going to happen is you risk dehydration. And if you become dehydrated, then that's a lack of energy, headache, dizziness, gets very dark, concentrated urine. Big warning signs. Again, your body is really now starting to scream, have a drink. And if you still ignore it for a prolonged period, then you can have damage to your kidneys and liver and even death. So not good to ignore those warning signs. If we've got an excess of water in our blood plasma, then what we find is that then our cells are going to have, again, that problem of potentially bursting. So the kidneys produce larger volumes of urine in order to reduce the water levels in our blood plasma to avoid this. Last thing we need to talk about are three terms they could use. Hypertonic, that means we've got high levels of glucose and salts. Hypotonic, low levels of glucose and salts. And isotonic is where we contain iron concentrations equal in the blood plasma. Right, that's actually gone through everything in biology and combined science, higher and foundation, B1, B2 and B3 for your exam tomorrow. So it was a bit of a whistle-stop tour because we only had 90 minutes to fit it in, which we have done. Obviously, if there are sections that we've gone through tonight that you are not 100% certain on, then by all means head on over to the channel and look up the relevant playlists. So go through those key areas, but don't spend all night doing this. At this point, you are going to know virtually everything you can fit into your brains before tomorrow. So hopefully you know quite a lot from what we've gone through tonight. If not, pick out those key areas that are more likely to come up because they're always popular on the old ones. Photosynthesis, enzymes, respiration, always favourite topics in the past. But I can't guarantee what's going to come up. It's a new specification. We've not seen a proper paper on this yet. When you go into the exam tomorrow, do remember to read the question carefully, because the number of times that I mark exam papers and it's quite obvious you skipped the giant chunk of information at the top which gave you the answer and went straight to the bit that was just above the answer line, is crazy. So read the question carefully because OCR do like to give you a fair number of answers in that. Do highlight key bits of information in there as you go through. As much as they like to say don't write anywhere but on the answer lines, you can highlight, it's fine. Just don't write in the barcodes, otherwise you're in trouble. Once you've done that, do go through, answer all of the questions. Don't leave a blank space. Worst case scenario, you get it wrong. There's no negative marking. We don't take marks off if you get something wrong, but there's a chance you got it right. Nothing else might provide a little bit of entertainment for the examiner marking your paper if it's truly off the wall and wrong, but try to keep it science related. Obviously, the multiple choice, there is only one answer, so don't write more than one. And if you've got weird B's and D's that look about the same for whatever reason, also circle it in the question just to make it really clear which one you are actually writing down. There's one 6 marker in your paper, which was denoted by the little asterisk. When you see that, you don't have to write in these long flowing paragraphs with all this flashy language they teach you in English. Scientists are marking your papers, folks. You can bullet point it. Use a capital letter, use a full stop, use a logical structure, done and dusted. You are not going to get marked down because you didn't write in a paragraph or use an adverb or whatever random stuff they want to tell you. Clear, facts, bullet points, absolutely fine. Obviously, if you finish your exam early, do go back over and double check it to make sure you've, one, not missed anything out, and two, answered everything to the best of your ability and that it makes sense is always good. Only other thing I'll say is make sure you do take a calculator with you, because we know there will be maths questions on there, and do read it, because if it says give it to two significant figures, give it to two significant figures, because otherwise you're throwing away one of those marks. Hopefully you found everything here useful tonight, and obviously we are going to be doing one for chemistry again in a couple of nights' time, so good luck in your exam tomorrow, folks. Obviously, my lot, I will see you tomorrow, and we can go through anything that you weren't sure on in the lesson. But other than that, best of luck for tomorrow, and hopefully see you again in our next live seminar.